Today we're going to be covering chapter 7 over metabolism of microbes, their catabolism, anabolism, different metabolic pathways of how they regulate energy usage and produce biomolecules. A little overview of how the chapter is structured. First, we're going to go over generally what is metabolism and what are enzymes and their role in biosynthesis pathways and catabolic pathways. Then we're going to move on to the pursuit of energy. So how do we gain energy and collect and harvest it, and how can it be used? Then catabolism of breaking down molecules to extract energy out, and anabolism and other pathways in metabolism of how do we actually, once we have the energy and the raw materials, how can we build them into larger molecular structures? So our first section on metabolism and the role of enzymes. Metabolism is the sum of all the chemical and physical processes occurring in the cell. And it comes in two varieties that are kind of the yin and yang of each other. You have anabolism, that is any sort of energy consuming process. So this would be a biosynthesis pathway. So maybe you're trying to go from just raw amino acids to a whole protein, trying to build things up, going from smaller to larger. Catabolism is the chemical breakdown of complex compounds into simpler units to be used in cell metabolism. So this would be more like if the microbe took in lots of different sugars, then they could break them all down into monosaccharides, and then maybe even further into even smaller units that could then be fed back into an anabolism pathway to build back up into something else. Often ATP or energy will be harvested through catabolism and then used in anabolism. Assembly of smaller molecules into larger ones is necessary for the general function of the cell. We need a wide variety of macromolecules, from lipids to nucleic acids, proteins, and sugars. And to do this, ATP, or adenosine triphosphate, the energy currency of the cell, is required. Degradation of biomolecules into smaller ones is catabolism. So going from large chunks and chopping them up into smaller ones is catabolism. And we can collect ATP in these processes and then spend them as energy to actually assemble larger molecules. To give you a little flow of how this might go in a cell, you would start with nutrients that are taken in by the microbe. So maybe they have a lovely tasty meal of glucose today, and glucose would be fed into a catabolic pathway of glycolysis, the Krebs cycle, maybe the electron transport chain, fermentation, all of which are going to yield energy. So whatever was taken in is going to get broken down, and along with it, it's going to yield energy. And we'll get some key molecules like pyruvate and acetyl-CoA out of them. These are going to be smaller than the molecules that we started with. And now that we have these raw precursor molecules and some ATP, we can begin building things. So you can go through anabolism using energy in these building blocks to go from very small basic molecules to individual amino acids, sugars, fatty acids, and so on. Further anabolism will get us macromolecules like protein chains or DNA out. And then if we go even further, using more energy and assembling them even more, we can get to a whole functioning microbial cell. Now, to do this, it could happen. These reactions are possible on their own, but they're very slow. And to catalyze or expedite the processes, we need enzymes. Enzymes are biocatalysts that increase the rate of a chemical reaction. They're not going to be a part of the reaction in that they're not products, they're not consumed by the reaction, but they can speed things up. Substrates are the molecules that enzymes act on that are actually a part of the reaction. So let's look a little bit more at what are some characteristics of an enzyme. How would we identify? Typically, enzymes are comprised of protein, sometimes RNA, and then occasionally they'll require cofactors, often more like metallic ions, to help them function. They'll act as catalysts to speed up the rate of a cellular reaction. They can't make a reaction that is unfavorable suddenly happen. All they can do is just expedite a reaction that was already going to occur, but would have been much slower. They have very specific, unique shape, specificity, and function. So since they're proteins or RNA, they're going to be folded in 3D and have a very defined shape, which makes them specific to specific substrates. So they're only going to act on certain molecules, not just on any molecule that happens their way. They can enable metabolic reactions to proceed faster, which allows life to be possible. They have an active site for a target substrate. So sometimes they're shown kind of as like a Pac-Man kind of shape. So they'll have an active site on the protein or the RNA where the target substrate can bind and be worked on, basically. They're going to be much larger in size than the molecules they're acting on, and they associate closely with the substrates, though they're not going to get sucked into the reaction. They cannot be used up. They just basically cycle. So they work on one substrate, spit it out, find a new one, work on that one, and so on. They're not going to be permanently used up. They can be recycled, so they function in extremely low concentrations again. So they work on one substrate, and once it's done, they move on to the next one and the next one, and so on. They are very sensitive to their environment, specifically to temperature and pH. So they have a narrow range of how hot or cold it can be, how basic or acidic it can be in order for them to function properly. This is largely because if you think back to Gen Chem, the charges on molecules change when the pH changes, which then changes the 3D structure. Or with temperature, it can cause degradation. So if it's too hot, then suddenly your protein has kind of folded a little wonky and is a little melted, and now it's not as specific as it once was. They can be regulated. So they're regulated by genetic mechanisms and feedback loops so that we're only making the enzymes when we need them, not just all the time. Though there are some that are on all the time. So they can be regulated. They're not always regulated though. So again, most enzymes are going to be proteins. Some are made of RNA. Holoenzymes are the term we use for the enzyme that is made of protein and has their cofactor. That protein component is known as the apoenzyme. So you think, okay, we have one apoenzyme with a coenzyme, cofactor. Those together form the holoenzyme or the total enzyme. Cofactors can either be organic molecules, known as coenzymes, or inorganic elements such as metal enzymes. This would include things like magnesium. That's a very common one I've seen with a lot of RNA enzymes. One example of a well-known enzyme is catalase. This is the enzyme that will break down hydrogen peroxide into water and oxygen. So it has a holoenzyme that is comprised of apoenzyme, protein component, along with iron as its cofactor. Okay, so a little picture to go with it. Holoenzyme is all of this. It's comprised of the metallic cofactor, like the iron in our last example, and the protein in purple. And there's a variety of different ones. You can have multiple coenzymes and metallic factors. I think if I remember correctly, I found one paper had example of an enzyme that had 6 kind of additional coenzymes and cofactors, but it's not like you're gonna have 100. It's gonna be like a couple, 1 to maybe 6 kind of is the range. Enzyme substrate interactions will occur at the active site. So there's going to be one spot on the 3D structure that's known as the active site where the reaction is going to occur. There's going to be a specific fit between the enzyme and the substrate because, again, we want that enzyme to only be acting on the one specific molecule that it's required to. Sometimes you'll hear it referred to as that it is a lock and key fit. So it's not like any old molecule that wanders in can be reacted on. It's just going to be that one protein that it's reacting on or that one sugar or so on. So you can see here, it's very specific. You have our enzyme in purple with multiple different molecules it could act on, but the turquoise and green one won't fit. It's not a lock and key fit, so it doesn't get reacted, whereas the pink and orange fits like a glove. So that one's going to come in and form an enzyme substrate ES complex. And then once it has acted upon it, you can see the products that are released. The enzyme expedited the process. A little bit more on cofactors. So these are the support staff, basically, for the enzymes. They can be metallic elements. They can also be organic, but they're micronutrients often. Metallic cofactors activate the enzyme and help them bring the active site and substrate closer together. So sometimes you'll see that they slightly change the 3D structure of the enzyme, but they assist in the substrate finding the active site and bringing it all close together. Coenzymes are the organic cofactors that work in conjugation with the apoenzyme to alter the substrate. So these are going to be the organic cofactors. You can also have metallic ones, but many of the coenzymes are going to be derived from vitamins, which highlights why there can be vitamin deficiencies that can really mess up your metabolism, because if you don't have the vitamins, then you don't have the coenzymes, then your enzymes don't, your holoenzymes don't function right. Oh, and now your metabolic pathways are all whacked out. Okay, so many of them come from vitamins that are gotten from the general nutrients that is provided to the cell. Enzymes are classified and named according to their characteristics. So what they do, what they act on, their name is going to give you a clue of what their job is. The enzyme name is comprised of 2 parts. The first part is going to be their characteristic. What's their job? What molecule are they acting on? What are they doing? And then the ending is always going to be ACE. So if you see a word that ends in ASE at the end, that gives you a hint, hey, this is an enzyme. So a couple examples, carbohydrates is if we take the first part of the word, carbohydrate digests carbohydrate substances. And we see the ACE at the end, so we know that it is an enzyme. Protease is, again, if we look at the beginning, starts with protein. Oh, that sounds like protein. So proteins breaks down proteins. As earlier mentioned, enzymes can be regulated. So some can be on all the time. Some can be on only as needed. So the as needed ones are the regulated, constitutive are on all the time. Constitutive are enzymes that are always present in relatively consistent amounts. So they're going to be the same no matter what's going on in the environment, regardless of the amount of substrate. If there's none or a bunch, either way, it's about the same. One example is the enzymes necessary for metabolizing glucose. Those are going to be pretty stagnant there all the time because you don't know when glucose is going to show up. You got to be ready at a moment's notice. Regulated enzymes, though, are enzymes that are turned on or turned off. So the turned on ones are known as induced enzymes. Turned off are ones that are repressed. So basically, are we turning them on only when we need them, or are we turning them off when we need them no more? So it's all in response to the changes in concentration of the substrate. So one example is the enzymes needed to make tryptophan. Tryptophan is an amino acid. So if we have a bunch of tryptophan, there's no point in making more of it with the enzymes that make it. So we can turn it off, and then once the level of tryptophan drops down, then we can turn it back on again. It's all about efficiency and using your resources wisely. Okay, so examples of constitutive and regulated. Constitutive would be, it's the same no matter what. Even if we add more substrates, there's no change in the amount of enzyme. Regulated is when we're kind of responding to the amount of substrate that is present. So if we add more substrate, then we're going to make more enzymes. Or maybe if we remove the substrate, then the enzyme gets repressed. We don't need to make as much. So this one is more fine-tuned than the constitutive enzyme. The cell's environment influences the enzyme activity. So not only does it influence it based on how much substrate is there, but also the general parameters of the environment affect the enzyme's operation. Enzymes only operate under the natural temperature, pH, and osmotic pressure of the organism's habitat. So there's a very defined range of, if you're an extremophile, then we really need to be operating and optimized for high temperatures. But maybe you're a human body pathogen. If you go beyond your optimal range of pH or temperature, then the enzymes become deactivated or denatured. When enzymes are subject to changes in environmental conditions, they'll become chemically unstable. So they may get folded wrong a little bit and will become non-functional. Low temperatures inhibit enzyme reactions, so they're going to slow them way down. High temperatures completely denature the protein enzyme. They make it all folded, all wonky, and then it can't function, completely cannot return from it. So if you were to put a microbe in the freezer, it would slow down the reaction, but if you pulled it out, then the enzymes would kind of come back. If I put it in boiling water and then cooled it down, the enzymes aren't going to come back from that. That cell is probably dead. Okay, so cold is much easier to salvage than hot. Denaturation is the process by which bonds within a protein are broken. So when we can change maybe the charges on certain amino acids, that changes the bond structure. We change the bond structure, we change the shape, and we know structure determines function. And if we change the structure, it's not going to function. There are two types of inhibition for enzymes: competitive and non-competitive. Competitive is a control process that relies on the ability of metabolic analogs to control growth by competing for the spot on the enzyme. So competitive is maybe you have an active site for glucose, and glucose is the normal molecule, but maybe lactose looks pretty similar, so it can come in and kind of compete or vie for that spot. So an example of that, I have the cars in the background here, is like if you're driving around in a really full parking lot, maybe you're trying to find a good spot in upper lot, I don't know, okay? You are competitively looking for that spot with other cars, and maybe somebody comes in and cuts you off and takes the spot. Well, they competitively inhibited you from getting into that active site because you guys both fit in the same parking spot. Non-competitive is when the enzyme inhibition involves binding to another site other than the active site. So if we think back to my parking analogy here, it would be like if you went to Walmart and someone maybe drove a shopping cart over the corner edge or something of your parking spot. Well, they changed the shape of your parking spot. They didn't take the whole spot. They just kind of shifted it a little bit to where now you no longer fit. They've inhibited your parking there. Okay, so both of them have the net result of inhibiting the reaction. They just do it in a different way. Okay, so pictures to go with it. You have two molecules that can both bind at the active site, the red one and the pink one. And red one's the normal one that makes the products, but the pink one can also bind there, doesn't make anything. And if they're both there, it's going to slow down the regular red reaction because the pink ones are taking up valuable space. Non-competitive, there's only one molecule that binds to the active site, this red one, but maybe the yellow one can bind on the side here. And this one changes the shape so that, like the shopping cart idea, so that then the reaction cannot proceed. Okay, so there are further controls of enzyme synthesis. Controlling enzymes by controlling their synthesis is another tactic. So enzymes, since they're made of protein and RNA, are made in-house by the cell, and they do wear out. So they have a defined lifetime, and eventually they're going to stop functioning very well. They need to be replaced. So at that point, when they can be replaced, we have the option of, do we need to repress them or do we need to make even more to replace them? Okay, so enzyme repression is a way of stopping further synthesis of enzymes. We're just saying, hey, we're not going to replace them. I know we had 10, 5 are worn out. We're just okay with the 5 now. Or we can have enzyme induction. This is a way of turning up synthesis through genetic induction. So maybe you're having some turnover, and instead of replacing 1 with 1, suddenly you're going to be replacing it with 5. And so you're trying to increase the number of overall enzymes that you have. So hence I have a picture over here. It's kind of like fine-tuning a dial, like on an audio mix. Basically, you have options or notches, not just an on-off switch. So you can kind of fine-tune as you need. Do I need 5 enzymes, 500, 20? You can kind of decide. Okay, so just to give you the flow of how we would repress a enzyme production, you start with DNA, with the gene that you need to make the enzyme. So the DNA gets transcribed into RNA, which then gets translated into protein. And then that protein gets folded up to form the enzyme structure. And maybe it binds with its cofactor. And now you have your holoenzyme in purple here. And it can act on the substrate. And then you get products as a result. Well, you make a bunch of these products. Well, these products can come in and bind to the DNA, which can inhibit transcription, which means we're not going to get any more of those enzymes, which means we've slowed way down or repressed the production of further enzymes. Because we have enough of that product, we don't need to keep making more of it.